really fortunate having Eric Schadt visiting campus again today. Eric has been one of the most creative forces in genomics for quite some time. Let me give you a little bit of brief background to his history. He was an undergraduate applied math and computer science major at San Luis Obispo, and then went to UC Davis, where he got a master's in pure mathematics, and then went to UCLA, where he got a Ph.D. in mathematical biology, or was it? Right. Yeah. Biomath, which required essentially passing 2 different qualifying exams in 2 different disciplines. So those of you that just went over this hurdle can recognize how unpleasant that might have been. Since then, his career has been among the most interesting that I've ever seen. He left UCLA and took positions at various companies, including Chevron, Silicon Graphics, and various others. But I first became aware of Eric's work when he went to Rosetta Informatics, one of the very interesting genomics companies in Seattle. And I have to say that during that time, Eric published more of the papers that I am most jealous never to have had any part of than anybody else I know. He really set the stage for how we understand the integration of genomic information with gene expression profiles in a function-driven discovery manner in organisms ranging from yeast to mouse to man to maize and everything in between. It's an astonishing series of papers, and I certainly recommend all of them to you, and we'll assign some of them to some of you in the coming years. Since that time, Eric has gone to what has excited all of us a great deal, probably the most innovative DNA sequencing company on the planet. The next-gen sequencing is fundamentally changing the way we approach biology in every conceivable way. And Eric, it's a delight to have you here, and we'll hear many, many interesting surprises.
Great.
Thanks, Eric.
And hopefully Jasper knows that it was, you know, when I was at Rosetta and everybody was wondering why the heck I wanted to do any of this, it was Jasper who was able to weigh in and tell them that it was a good idea.
You have a cell phone on perhaps? Because I'm hearing that.
Are you? Yep. Okay.
Thank you. And then I'm getting you off the podium, Mike, so I need you to stand in that vicinity, and I'll be so happy with you for the rest of the day.
All right. Geez, there's a lot of rules here. Yeah, don't mess up. So we're all pretty amazed in the age we're living in with these technologies coming along, enabling us to take amazingly comprehensive snapshots of these living systems over many different dimensions. So not just the DNA dimension anymore, but many other dimensions. And what's been striking to me on that is how we still, people like to stick to what they're good at, so we still tend to take this one-dimensional view, even though we have these massive amounts of data types available to us. And I like to highlight the GWAS studies, these genetic association studies, as one example of focusing on a DNA dimension that's delivered some pretty interesting discoveries. And we saw in 2007 how the genetic variation, the mapping of common variation and its association to disease was discovery of the year. And we saw these personal genome companies coming on the scene that were going to tell us all how to live better lives by genotyping our DNA, and we saw that type 2 diabetes was decoded. So it was sort of all the rage, and the idea, of course, of looking at this single dimension is pretty simple, right? It's you're looking at variation in the genome that's varying, right? So you have these SNPs or whatever the marker is, and then you're forming this simple correlation between changes at a given marker and some disease trait or phenotype that you care about. So it's a pretty point-wise, you're going through each one of these SNPs and you're performing this test that you learned in high school to associate to disease. And it's interesting because it does give you some insights into the causal drivers of these phenotypes. But what it's not the DNA directly, right, that's causing changes in these phenotypes. It's acting through some intermediates, and it turns out that intermediate is all the rest of biology, right? So it's all of these processes going on that are defining how changes in DNA are impacting the molecular states in these networks that we all know to be true, that go on to change their state and goes on to cause changes in phenotypes that we care about. So, the GWAS studies are ignoring, 99% of the information, the biology, and then, of course, we see the papers in 2009 coming out that they're ignoring 99% of the biology and then wondering why the GWAS studies aren't being more successful. And they're successful from the standpoint of locking onto loci, but it wasn't as amazing as everybody thought it would be, right? It wasn't explaining all the variation in these diseases. It wasn't leading to amazing drug targets and that could impact human health. And so the aim of this community, which I really equate, and I shouldn't even say it because it's being videotaped, but the human genetics community, they remind me of drug addicts with attention deficit disorder, right? Because they spend all this money in a very short period of time to understand the data, and the answers aren't all falling out of the sky. So now they're running over and they're going to sequence all the exomes, and that's going to solve all the problems. And my point would be that we have to be integrating, we have to embrace the complexity, go beyond this one-dimensional view, right, of understanding biological systems, and integrate that GWAS information with all the other kinds of information that can be generated to understand these systems. And so I like showing this. Got this from Gary Churchill a long time ago, but it well illustrates, right, the childhood story you heard about, that if the blind man is sensing a given object and just looking at one dimension, he's going to walk away with a very different understanding of what that thing is than if he's sampling the entire thing. And so but to embrace that kind of complexity, our minds are going to fight us every step of the way. And the reason our minds are going to fight us is because our minds are wired to tell stories, not to understand statistical uncertainties. And we see this throughout history, right? Whether it's coming up with the story of Zeus, the sky god, to understand lightning, or coming up with a story about how the Earth is the center of the universe, or that the Earth is flat, or that biological processes are driven by simple linearly ordered pathways. These are the kinds of stories we like to tell. And but how do we fight the want to tell stories? We fight the want to tell stories through technological innovation and application of that innovation. So whether it's Galileo and the telescope resolving mysteries of the universe, or the new PacBio machine that again is going to offer unprecedented snapshots of systems to see much more of the system, to put together the complex circuits that we know are underlying the complexity of the phenotypes like disease processes. Yeah, probably. Yeah, that's a possibility. So in embracing the complexity, the idea is to understand the flow of many types of data types and understand the flow of that information. So whether obviously the DNA variation and the interaction of that DNA is an important source of information, but so is the other types of data that are affected as a result of change of DNA. So whether it's change in DNA leading to changes in these molecular states, whether RNA, protein, which go on to affect the cellular processing, which go on to affect tissue level behavior, and on up to the organism level, where the physiological states we associate with disease are affected by the cascading of that information. That's the kind of flow we want to understand. And to understand that flow, we obviously have to be understanding all the different data dimensions and trying to understand them in real time, right? We're dynamic systems, and so we're not these static beings, we're dynamic systems, and understanding the nature of these data in the sorts of ways that actually occurs in your cell hopefully convince you that that's important. And so I'll first sort of try to go quickly through this technology and how it's going to revolutionize our ability to capture these different states of information and in real time before I get into the more fun science of the application of that. And the idea of the technology that I'll talk about from PacBio was centered on being able to observe. So we're interested in sequencing the DNA, obviously, and the second-gen technologies are sort of these very over-engineered systems that are leveraging DNA polymerase as a reagent in the sequencing reaction. And what the Cornell group said is, well, this DNA polymerase is actually a sequencing engine, right? So we don't want to, why exclude that from the possible sequencing things we could develop? If we could just observe that polymerase in its natural state as it's sequencing DNA, well, we'd have a pretty amazing sequencing engine, because that engine is moving at 750 bases per second compared to sort of state-of-the-art second-gen technologies that are moving at 1 base every 50 minutes. It's very processive, right? It's generating strands that are tens or hundreds of thousands of bases long, so it's not a 75 base pair read, it's a 100,000 base pair read. And of course, it only requires a single molecule to carry out the reaction. So it's pretty amazing if that could work. And of course, the hard part of that problem is how the heck do you look at a single molecule of something performing its activity when the sort of state-of-the-art of the microscopy that existed before PacBio was that you'd be looking at lots of other things in that volume of information in addition to the single molecule. So how do you sort of focus in on the single molecule? And the thing they came up with, the very clever but simple design based on this waveguide transmission below cutoff, was to simply etch these tiny holes, what they call zero-mode waveguides. So you take a thin metal film, affix it to a solid substrate, and you're etching in these zero-mode waveguides, sort of all fancy terminology for their tiny little holes. So the tiny little nanometer type holes that are etched in there. And the beauty of this type of design is the diameter of that hole is smaller than the visible wavelength of light. So when you bounce light up, no standing waves can exist in this waveguide. That's why it's called a zero-mode waveguide. And instead, the light hits and exponentially decays into the hole and only illuminates the very bottom portion of the hole. So in fact, it's only goes up to like 10 or so nanometers. And so the confinement volume here that's illuminated is 10 to the minus 21 liters, a zeptoliter, which I didn't even know what a zeptoliter was until I was working at PacBio. But it's an amazingly small confinement volume that has the ability to highlight a single molecule without illuminating everything else that's going on in the mix. So when you think of the signal-to-noise problem and you want to be looking at a single molecule, you want just that single molecule illuminated without everything else that's going on. So it has extraordinary signal-to-noise, this type of setup. And this type of technology, it's what exists in your microwave, right? If you ever look at the inside panel of your microwave, it's a thin metal film with tiny holes perforated into it, and that allows, those holes are big enough to allow visible light to come through so you can see the food being cooked, but the holes are small enough to where the microwave radiation cannot penetrate, so it just bounces around. So your face isn't getting melted off as you're watching your food getting cooked, and I like that feature. It's a cool, glad they put that on there. But it's the same simple idea, but now we're just down to the nano size to make it work. So how does this actually work? This is single molecule of DNA polymerase anchored into the bottom of that hole, latched onto a single strand of DNA. And of course, we have to illuminate that, so we need something that makes that visible. So what's done is the dyes are tagged, or the nucleotides are tagged with a dye. That can be, when the laser hits it, is energized and emits photons, so you can see the nucleotide that's being incorporated. And importantly, that dye is on this triphosphate linker, not on the nucleotide itself, like all other second-gen technologies. The importance of that is once the nucleotide is incorporated, so when it's being incorporated, you want the dye there because you want to see the event. But after the incorporation, you want that dye to go away, because if that dye was there and the other nucleotide came in, you would start seeing multiple signals and you wouldn't be able to understand it. So being able to stably put that dye on that triphosphate linker, critically important, because if it was on the nucleotide like the second-gen technologies, the polymerase would not like that. It would ultimately just get gummed up and halt after a couple of nucleotides, and it wouldn't be able to do the sequencing. So when we look at this in fast real time, here's what it looks like. Remember, only the bottom portion is illuminated, so when nothing's being incorporated, you have pretty flat signals across the four colors of representing the four different nucleotides. And these nucleotides are randomly diffusing in and out of that hole in the microsecond regime, so way too fast to be held in that confinement volume and actually seen by the cameras. So you don't really see that. You see that low-level buzz. But once the polymerase latches on, finds the nucleotide it needs to incorporate in replicating this sequence, now this is a millisecond event. So that's slow enough to be seen by the cameras. You see the signal bounce up over a sustained period of time so that you can actually see what nucleotide is being incorporated. And then after the incorporation, again, this dye is going to float away, and you're going to see the signal bounce back down to zero, and the enzyme will translocate to the next base, and you'll be able to see the next base being incorporated. And so what this looks like in real time is then something like this, where each nucleotide that's being incorporated gives you a color burst of light so that you can tell which nucleotide is being incorporated. And that's real time. This thing's moving at 3 to 5 nucleotides per second, incorporating those different bases, and you can watch that occur in real time. And just so you can see an actual movie on that, here's a movie of, so that was one zero-mode waveguide you were looking at, so you can multiplex this by looking at thousands of these simultaneously. And this is a cross-section of a chip 100 microns wide. That's about the width of one of your hairs, and there are 3,000 zero-mode waveguides there. So 3,000 DNA polymerase molecules sitting there waiting to sequence their DNA. And when we activate this movie, you'll see it go black. That's the laser's off. When the laser fires on, you're going to see these holes start to sparkle. So they're not sequencing right now. They're in a fixed state waiting for the cation to be introduced to carry out the sequencing. When that cation is introduced, you see all those different flashes of light. Each flash of light is a nucleotide being incorporated in real time. You're observing that in real time over thousands of reactions all simultaneously. And that's an amazing invention, right? To be able to look at 3,000 individual molecules performing their action in real time is just unprecedented. And this is, this goes beyond DNA sequencing, right? Because you could have lots of things in that zero-mode waveguide, lots of proteins that you would want to observe. And so I think it's a revolution generally in protein biochemistry because of that single molecule real-time resolution. And again, it doesn't, the DNA polymerase was just one kind of molecule to have in there to drive, to get funded, right? To have a product that could come out and make money. But there are lots of other things you can put in that hole, right? You could put reverse transcriptase in that hole and look at direct RNA sequencing, which we're actively doing. So that'll be coming out as well as the DNA sequencing. You'll be able to do direct RNA sequencing, so without having to go through the creation of cDNA and so on to assess transcription. Of course, there was just a paper by Jody Puglisi at Stanford in Nature on anchoring ribosomes. So having ribosomes in those holes and looking at the real-time translation of mRNAs. And so here's one of the figures from that paper, right, where here's the mRNA. It's an artificial construct with the alternating FK amino acids. And so here you're observing directly by tagging the tRNAs the real-time translation of that mRNA as it's going through the ribosome. And again, just unprecedented ability to look at that type of information in real time, not only getting what's being translated, but the kinetics behind it, which I hope to show is pretty important. So I sort of lied on my little talk abstract. I'll talk a little bit about the hydrogen production problem as a motivation for how to use this information, but I'll also give an example on the human space. But on the hydrogen part, so the first thing I wanted to do with this type of technology was to look at lots of different strains of bacteria that naturally produce hydrogen gas, given the importance of hydrogen as a big commodity chemical and potential alternative fuel source. So I partnered with Carrie Harwood at University of Washington on looking at Rhodopseudomonas palustris, which naturally produces hydrogen gas under photosynthetic processes, as a result of photosynthetic processes. And so the idea was, let's sequence lots of different strains of this bacteria that are producing hydrogen gas. It highly varies between the strains how much hydrogen gas they produce. And let's sequence the DNA and RNA in all of those bugs and see if we can't put that information together to understand the networks that are modulating the hydrogen production and whether we could tweak those to make the system more efficient. I started this this project on using Illumina sequencing back when the 32 base pair paired-in reads were sort of state-of-the-art. So it was about a year and a half ago. And so generated 19x coverage over all of the strains. And I'm going to show you an example of one of the strains where we were unable to assemble the genome from that short read data, which a big surprise, right? So what we ended up getting were thousands of short contigs and N50 of around 1,500 bases, almost completely useless for playing the kind of genetic game I wanted to play, for looking at how the structure of the genomes were changing over these hundreds of bugs to change the structure of the network. So we wanted to see whether if we layered in the longer range data that could be generated from the PacBio technology, whether we could rescue that type of experiment. So again, we started with the full, just the Illumina data, short read data, where we were using Abyss to do the assembly. We're using, there are other programs like Velvet and others that we could have used, but what we saw with those programs is they were really misleading us. The contigs that were coming out were not very accurate, and when we tried to layer in our data, there were just too many mistakes. So we biased ourselves towards generating highly accurate but short contigs before we layered in the long-range data. So we had, from the Abyss assembly program, we had 6,000 plus contigs with an N50 of 1,000 bases, which is pretty sad. And then we layered in the long-range continuous read data from PacBio. We also layered in the strobe sequencing data. So this is sort of you can think of it as a mate-paired strategy where we can strobe the laser to have less time with the enzyme under the influence of the laser light. So what happens, why the enzyme doesn't produce in this system long reads, is the enzyme eventually gets killed by some event of the dye transferring energy to the enzyme instead of emitting photons. And so we get this point event that damages the enzyme, so we can just turn the laser on and off and let it run through the molecule to get longer and longer reads. So we sort of generated both these continuous reads, which were averaging 1,000 plus nucleotides, plus the strobe reads, which were going into the thousands of bases. And at the end of the day, got that assembly down to 12 contigs with 13x coverage of the PacBio data, 19x coverage of the Illumina data, and 620, an M50 of 620 kb. A very, very successful assembly. Interestingly, at the same time, and this is just showing you the final assembly and some of the statistics around, again, what we started with versus what we got when we layered in the long read sequences. Interestingly, at the same time, one of these bugs, of the couple hundred we sequenced, one of them was being sequenced by JGI, and they deposited this in the public domain when we were doing our sequencing. They used Sanger sequencing on this, came up with an assembly that was comprised of 58 contigs. So we looked at what would happen if we layered in, again, that long-range data we had generated. Could we improve this assembly? And by layering in the long-range data, we got that assembly down to a single contig. And again, the advantage of that is there was no manual finishing, right? This was completely automated, just generating the long-range data, putting it in the assembler, and getting a single contig versus going in with, as people know who have to finish genomes, a very laborious task of going in there with a manual finishing. So seeing that, we think the long-range data is going to be a great utility for assembling genomes de novo. So I should have said that initially. These are all de novo assemblies because we don't have references for all the different bugs. Their genomes are changing very radically. But the most interesting thing to me wasn't the static sort of DNA view of the genome. It was leveraging the real-time nature of this information to, say, go further with the interpretation of the DNA. So remember, all of these different assays that can be run on that machine, they're happening in real time. So you're looking at the DNA as it's rolling through the DNA polymerase. And what you can see is that variation in the kinetics through that enzyme is highly correlated with chemical modifications that we see on the bases. So for example, here's a strand of DNA being sequenced by a polymerase. The only difference between these two strands is this A residue is methylated in this strand, but not this strand. And look at what happens when that DNA polymerase encounters an A that's been methylated. Look at the time it takes to translocate to the next base. It's much longer than when that A is not methylated. So this type of variation in the kinetics we're seeing as correlating with all of the different types of modifications that we know of, whether it's a C methylation, this methyl A, whether it's different types of DNA damage like 8-oxy-G or other types of things. They all have signature pausing events that we can pick up on. So I decided to take this, this was work that had happened right around the time I was coming to PacBio. So I decided to take that a step further and say in these populations, so we're sequencing across these populations of bacteria, and what we can do is actually start leveraging, detecting this kinetic variation over the population as we have high fold coverage on each site in each individual strain over a population of strains. So the idea is to look at, is the variation in the kinetics over a given site, regardless of whether we know or not what type of modification could occur, are we seeing differences in variation going on? And so we can pretty simply model that. Since these interpulse durations, the time it takes the enzyme to go from one nucleotide to the next, are exponentially distributed, we can come up with pretty simple mixture models that will actually just look across that site over the population and say, are there multiple exponential distributions represented here? And not only that, we can pick up the mixing proportions as well. So for example, for a methylation event that causes kinetic variation, we can assess from the population or a given strain whether 10% of the sites were methylated and 90% were unmethylated, for an example. So we can estimate the proportion of those distributions in the data. And since the effects of these modifications aren't constrained to the site, right? They have local effects over many sites. We can actually go a step further and come up with in fact, using now a conditional random field to take into account this correlation over the sites to extract more information to detect these sorts of things. And so I won't go through that one, but just to say that, so again, the game is we're going to look over all these bacteria, and we're going to look over the genome, and then at each site, we're going to apply that model and see whether we detect these variations in the kinetics. And then we may not even know what it is, but it's not important to know what it is. What's important is to track it as a covariate and see whether it's changing with other traits that you care about. And so we saw things like this when we did that over the whole genome. It was pretty cool. So what we're looking at on the y-axis is just the ratio of rates in fitting this model. So if there's kinetic variation going on, then the ratios of rates should deviate significantly from 1. So we see here whole regions of the genome that effectively have no kinetic variation going on, whereas other parts of the genome, we see all kinds of kinetic variation going on. And this particular region is enriched for genes that are involved in nitrogenase activity, that are involved in the hydrogen production process. So these things are not only representing kinetic variation, but that variation is correlating with the activity of the genes involved in the hydrogen production process. So we're right now in the process of validating what are those, what's causing that variation, and is it causal for the changes in the gene activity. But again, the beauty of this is it's completely data driven. You a priori do not need to know what, that it's a C residue, because that's the residues we know are methylated. This is completely agnostic to what the actual mechanism is. You're just detecting the effect and then seeing whether that effect associates with genes and phenotypes that you care about, and then you go in and try to understand what it is. And that's a complete revolution. It brings this time dimension to the DNA sequencing process, which has never had to be considered before. And given all this variation and how it's correlating, it's sort of taking the DNA information into an analog dimension, right, away from this digital AGCT domain and into this analog dimension. So once we have all of that kind of data, so I'm going to sort of pop in, I think now to how we integrate all this information to build a network. So we're grabbing lots of information. So you're looking at the DNA and the RNA, and we can throw in other types of information that were collected from other types of technologies, where it's DNA protein binding or protein interaction. So how do we bring that all together to build networks that are actually predictive of the future states of a system? So the main thing is how do we infer relationships between these genes, and in particular causal relationships. So if we sample from a population, whether it's a population of bacteria or humans, we can sample from that population 2 traits that we care about, and we can see that they're correlated, right? We can test for correlation because we see over the population, we see them tracking together, and the traits are related. And we've seen that this is pretty useful in the gene expression space, right? We've seen lots of people here and others have looked at patterns of expression that are highly predictive. So these correlation relationships are highly predictive of clinical states. So for example, in this breast cancer work we had done a long time ago, patterns of gene expression in the breast cancer could well discriminate breast cancer patients into good and poor prognosis signatures, right? So that's pretty useful. But the problem with this is it doesn't get at the causality of what's actually going on underneath. We don't know in this pattern what the drivers of that pattern are versus the responders of being in that disease state. So we can't distinguish the cause from the effect. And so we're sort of left in this situation where we're trying to make up stories again, where Brian was told by his doctor that fat masters are correlated with fat dogs, and so he's setting out to solve his weight problem by exercising the dog. And so it brings us all back to the right, correlation does not imply causality type arrangement. But it was amazing when you showed the biologists, again, it's the want to tell stories. I remember in the early days at Roche and other places where people would look at these patterns and be so amazed, and then they'd find their favorite gene in there, right, correlating with everything, and then they'd say, hey, that's the, that I know about that gene, it's involved in this process, we're going to spend $100 million, we're going to design a drug, and then it fails to have any effect because they didn't really understand the cause-effect relationships. So how do you typically establish causality? Well, in your labs, right, the classic approach is you artificially hit a gene and see whether it has the effect you predicted, right? So whether it's a gene knockout, a transgenic, an siRNA, or a chemical perturbation, you see that these 2 traits, now we're in a time dimension where you're looking, say, at a given animal or individual, and you're seeing over time that these traits are correlated, and now you have some hypothesis, right, that one is causal for the other. So what do? Well, you set up the experiment, and you now want to perturb one gene and see whether the other gene responds just as you predicted. And so when you do that, you punch that gene, it has the effect, and then you see the response in the other gene, right? So you follow that pretty simple line of reasoning then to conclude that this trait is causal for that trait. And so we can leverage that same kind of information but now use DNA variation as the perturbation source. So in the DNA variation information, we want to look at the different genotypes of individuals, stratify them by genotype, and now ask whether the changes in genotype, which is a naturally occurring perturbation, right? It's a variation in the DNA that we can see here is associating with the levels of expression or trait levels of the different traits. So this genotype is associated with low expression, medium expression, high expression, and the same thing with this blue trait down here. So because the variation in the DNA is tracking with both of these traits, well, now we're in a position to play a pretty easy game, which is to resolve how those two things are related. So again, we're starting with DNA variation correlating with two phenotypes. So we start with that information. If that's the starting information, then there's effectively only three ways those two genes can be related to one another. Either you punch, or either the variation in the DNA causes changes in this blue trait, goes on to cause change in the other trait, or vice versa. The DNA variation causes change in phenotype 2, affects phenotype 1, or that DNA variation is independently affecting both of those traits. So we can mathematically model those situations and based on the data, pick the model that's best supported by the data, and from that make the causal inference. But the bottom line is it's working just like your artificial perturbations, but this is naturally occurring, it's in a multifactorial context, and it's in a context in which a disease actually manifests itself, so it's a highly relevant context. But we're not just interested in establishing relationships between two genes or two traits, right? We want the network. And so to get the network, what we're looking at here are we have a number of DNA variations, right, all over the genome that are correlating with lots of different intermediate traits, gene expression traits or protein traits. And then we have other traits, clinical traits, for example, that we're actually interested in. So we have this sort of situation set up. Here's the big mess of things, and now we want to see how do we actually establish relationships between these guys. Well, how we do it is we systematically go through and test all pairs of genes with respect to the DNA perturbations, right? So we're testing using this causality type argument to go through and assess those relationships, and I'll just sort of speed up the search. And as we're systematically going through, we can actually construct the network of interactions over the entire space. And this isn't just an interaction network. This is a probabilistic causal network that gives us causal information. And then we can take those once, when we have this molecular predictive causal network, we can now integrate the clinical traits and see, well, how do those guys fit in? And so we can systematically carry out the same thing, and the resolution here is forcing this off the screen, but I think I can pull it back. So we can understand how these guys are related to the network of causal interactions and the molecular traits. And when we just focus on the genes and which ones may be causing the clinical traits to change, we can use this network to actually make those kinds of predictions. And that gives us then the hypotheses to go in the lab and actually figure out what's going on. And this, this is all Flash animation, and it doesn't play well with Microsoft, so I have to manually switch here. I don't know why these guys can't all work together. So we played this kind of game in the hydrogen example I was showing. So remember, we had hundreds of strains, DNA, RNA sequenced or profiled. And now we're going to integrate both the Illumina and PacBio data, that DNA variation data. We had expression data that was showing pretty nice patterns of expression that with the highest hydrogen producing strains had a certain pattern compared to the other guys. We want to integrate that information, just as I was showing you, to build networks that explain the hydrogen gas that's coming out of these bugs. And so now what I'm doing is we reconstructed that network, and I'm showing you a projection of that network onto the genome to highlight the importance of understanding the entire genome, of having the entire assembly for this genome. So what you're looking at here is a network where this cluster of genes right here are involved in nitrogenase activity, the core reaction that produces the hydrogen. But then you see all these other clusters of genes are causally affecting this particular cluster. So that's what our model predicted. And so what's interesting about this is you can see they're not randomly scattered all over the genome. They're clustered into groups that are occurring in common regions that may be under common regulatory control. Again, you need the complete genome sequence to see this kind of structure and to play the kind of game where you're looking at structural variations or rearrangements or orderings of genes as the perturbation to use to put that network together. And so in this particular example, we had this complex that was being affected by all of these different genes, and the genes in the red were known to be involved in hydrogen production. The genes in black were not known, but predicted by our model, and these are all sort of going on being validated, and several of these have validated, and we'll have a paper coming on that showing that these predictions were actually holding up. I also wanted to show how the same type of thing could be used in a human context, because it shows a greater complexity of the models we can pursue with this type of information. And now I'll show you an example where we applied this in a mouse cross, where now we're looking at 1,000 animals, and we're not looking at a single cell like bacteria, but we're looking at multiple tissues from each of the 1,000 animals. So hypothalamus and liver and islets and muscle and fat, and we're collecting those from each of the animals. And then we're profiling, genotyping, and we're going to integrate all this information to build networks that explain type 2 diabetes in this population. So this population is segregating a type 2 diabetes phenotype. So what we want to do, though, is we're interested in the system, not in a single tissue or a single cell. We're interested in how that system's communicating to give rise to type 2 diabetes. And I have like no clock.
Quarter to 15 minutes.
15 minutes? Well, I'll try to finish it up quick then. So the idea is, can we understand how gene information is flowing between tissues? So I'm not going to tell you how I humanized the mouse brain. That's a pretty fun story, but to save that for another day because we're out of time. And so we want to see how genes that are changed in brain in response to a genetic perturbation can cause changes in other tissues. And so here's an example of looking across multiple tissues. Here's a subnetwork that's explaining interactions between hypothalamus and adipose in that particular population. So the red nodes are monitored in hypothalamus, the green nodes are monitored in adipose tissue, and we're drawing edges between the nodes only if there's interaction between the two tissues, right? So all of the within tissue covariation we're throwing out, and we're only looking at the between tissue interactions over this population. And what we notice are these blobs, these highly interconnected sets of genes, coherent, but they're interconnected between the tissues, not within the tissues. So very important. What's most fascinating about this is when we started looking at these cross-tissue networks in relation to the individual tissue networks. So we reconstructed the networks for adipose independently of the networks for hypothalamus. In this type of view, all you have to really know is that these red boxes are the highly interconnected sets of genes, the subnetworks that make up the individual tissue networks. And when we looked for these highly interconnected sets of genes in these individual subnetworks, they were not represented. So these networks, invisible, if you're looking a single tissue at a time, you do not see them. You only see them when you look at the tissue simultaneously in the population. So entire sort of areas of biology sort of not being hit if you're looking a single tissue at a time. The only way you see these networks is multiple tissues at a time. So in this population, here are the kinds of circuits we can draw, right? We have liver, adipose, muscle, hypothalamus, and so on. And all these different red boxes now indicate different subnetworks that are operating either within or between the tissues. And we're drawing edges. What we're interested in, again, the boxes represent sets of networks, and we're interested now not in single genes, but how perturbations in a network can cause changes in other networks, either within or between tissues. And look at what happens when we start looking at the interactions of the subnetworks. So on the outside of the circle, we're looking at connections between these boxes, if there's interactions between the subnetworks within a tissue. On the inside of the circle, we're looking at interactions of those subnetworks between the tissues. Look at the connectivity on the inside of the circle, much greater than it is on the outside of the circle. But again, you don't see that if you're looking a single tissue at a time. So how that system is put together, functioning, how changes in one part lead to changes in another, you're not getting that picture if you're looking at the system one dimension tissue at a time. So you're missing a lot of the biology. And so what we had done is shown several of these networks that we observed in different kinds of tissues. Here is a network, what we call the macrophage-enriched network, that was, we called macrophage-enriched because it was concentrated with genes that were thought to be specific to macrophage. And they were also hugely enriched for genes that we predicted as causal for all the different metabolic traits that were being scored in this cross. So for example, genome-wide, of the 25,000 plus genes in the mouse genome we represented, we predicted 157 of those would be causal for atherosclerosis in that population. Well, 117, 75% of those fell in this single subnetwork. An amazing enrichment. The p-value is 10 to the minus 100th that occurred by chance. And when we initially did this, people were asking, well, how the heck could this macrophage network be causing all these different diseases? That doesn't make sense. But we started looking at how perturbations in this network were affecting other networks within the system, both within and between tissues. And what we found was perturbations in this network were affecting angiogenesis-enriched modules and vascularization-enriched modules and cell cycle modules. So we just had a paper that came out that showed how perturbations in this module affect the cell cycle module in the pancreas that limits the ability of the pancreas to proliferate beta cells to meet the insulin demand. So changes in this network are causing changes in that network which limit the ability of the pancreas to respond to that the high glucose, so needing insulin to meet that demand. And that's going on to cause type 2 diabetes phenotypes in that cross, right? So these, again, the system is highly connected, and we need to model that. So that particular network was found in mouse, but we actually want to treat these diseases in humans, because I learned at Merck that mice have no money to buy drugs, and Merck doesn't care about the mice getting well. They like making them sick, playing around with them, but they don't care about them. So what we have here, so we do the exact same thing we did in mouse, we did in humans, where we have these populations of humans where we have multiple different tissues represented. So we're playing the same game in humans where we isolate the DNA, do the genotyping, isolate the RNA, do the profiling or sequencing. And we have some of these populations have 8 tissues represented. It's amazing. This guy I'm working with at Karolinska Institute, we have 8 tissues, carotid, coronary lesions, atherosclerotic arterial wall, atherosclerosis-free artery wall, subcutaneous fat, liver, all from the same patients. That they open these guys up for open thoracic surgery, and they're just snipping away at these different tissues, which is good for us because we have cool data that we can show. And so the idea then is to take all of the gene expression traits that in humans associate with SNP. So we get these eSNPs, right? So eSNPs are SNPs that are strongly associated with the expression of genes in one of these tissues, and we want to use that information to reinterpret the GWAS to see if there's something more informative we can say, both functionally and missing heritability-wise. So we start with all the GWAS data. We're filtering that. We're going to filter that through this kind of funnel where we're first going to restrict to eSNPs. So we're going to say of the hundreds of thousands of SNPs that are typed into GWAS, if we restrict to just the eSNPs, are they enriched for associating with the disease? And then we're going to further restrict to eSNPs that are associating with expression traits that fall in networks that we predict are highly causal for disease in mouse, and whether those in humans are enriched for disease. So here's sort of the flow. We took the DGI study, one of the biggest studies for type 2 diabetes, big GWAS studies in type 2 diabetes. We took the hundreds of thousands of SNPs. We tested them for association to type 2 diabetes at the 0.05 significance level. And surprise, surprise, roughly 5% were shown to be associated with type 2 diabetes. So no signal, completely in the noise. Kind of what we'd expect given the high level of testing, number of SNPs tested. When we restricted to eSNPs that were associated with expression of genes in any one of these tissues, and then tested whether those were enriched for type 2 diabetes, we saw a marginal increase. So it was statistically significant, but not amazing, right? It wasn't like, wow, the lights really came on. So we said, well, what if we restrict to tissues that are more relevant to type 2 diabetes, like omental fat? So when we restricted to eSNPs that were associating with the expression of genes in humans that were from omental fat, we saw another significant, statistically very significant increase, and again, now a more interesting one, but still wasn't like super amazing. So now what we're going to do is restrict to those eSNPs that are enriched or that are associating with genes that are in that causal network I showed you that's highly causal for those metabolic traits, and see whether those are enriched for type 2 diabetes. And now that jumps up to over a 7-fold increase in the number of SNPs that are associated with that particular network I showed you in mouse, but in human are associating with genes in omental that go on to associate with type 2 diabetes. 40% of the genes in that mouse-based network associated with SNPs in humans that in turn were associated with type 2 diabetes. It's pretty amazing that what that's showing you is that this network is capturing all of that variation information, both genetic and environmental. Its state's being altered, and it goes on to cause the susceptibility to type 2 diabetes. So this is the thing that's showing us that it's not 50 or 100 genes that are involved in type 2 diabetes, as the GWAS studies are showing from the stringent thresholds. It's showing you that there are potentially thousands of genes that can be subtly affected by DNA variation and environment, but they organize into these networks which you can assemble and see them capturing the information and dramatically increase your power to establish relevance to the disease. So I'll sort of stop there and just say that we went on to validate several of these genes in the network. Again, this network was interesting, so we found genes that were highly predicted as causal in mouse. They were associating in humans with SNPs that were associated with type 2 diabetes, and that's an overwhelming amount of evidence that these are actually involved. So we go on and actually test those in animal models and saw here differences in glucose tolerance, just as we predicted. There were changes in glucose levels, insulin levels. All the type 2 diabetes traits we predicted were changed by this gene that was completely missed by the GWAS studies, right? Because the effects were too small to be seen with the population sizes they had. But by using this integrative approach, we pulled out this and hundreds of other traits that now can be considered as therapeutics in that space. And so just finish with saying that the main ideas here were one of integration, right? So I started by poking a little bit of fun at the genetics community just for their strong focus on DNA, but ignoring a lot of other information. And it's that other information that's responding more proximally to that DNA variation to cause disease. But these intermediate phenotypes, these are not acting in isolation, right? They're acting in networks, right? There are a lot of them, and they're interacting with one another. And so it's really the networks, the assemblage of these guys into networks that are responding to constellations of SNP changes or other DNA changes and environmental changes, altering the state going on to cause diabetes. And this really gives us a shift in thinking back to maybe 50 years ago or so, where remember the days when physiology really dominated, pre-molecular biology revolution dominated our how we study disease. So we use physiology, the study of physiology, to understand these disease processes. And when molecular biology came onto the scene, we sort of thought, oh, we're going to understand the individual genes and proteins, and we don't necessarily need physiology because we're going to understand the individual function and we're going to target them with small molecules and we're going to treat disease. But this has largely failed, right? In the pharma industry, this approach has largely failed. There are some successes, but the pharma companies are in today are the massive failure of looking at a single protein and inhibiting its activity or activating it and thinking that's going to cure or treat a disease has led to astonishing failure after astonishing failure. And the reason for that is you don't understand the context in which that change is occurring and how it's affecting other networks, how it's affecting other tissues, how it may give rise to adverse events, how the system may compensate for that hit to give you no efficacy. And that if we want to really understand how molecular biology impact clinical medicine, then we need to bring physiology back into the picture. And the way we bring physiology back into the picture is this system level view, that we organize that molecular biology information into these networks to understand the functional units that are underlying the physiological states that we associate with disease. And that's how we're going to have molecular biology have an impact on clinical medicine. And so I just want to thank my group at Genetics that did a great majority of this work. They're now transitioned out into Sage Bionetworks in Seattle. I'm still involved with that effort. The PacBio team on that technology, I of course had nothing whatever to do with the development of that technology, but I will get to be a benefactor of it. And then the different academic groups that were instrumental in forming the populations that we used for this study, both the bacteria and the mouse and human. So thank you.
I'm sure Eric will be happy to answer some questions, but before that, let me remind you that thanks to the persuasive powers of Susan Marquesi and the resulting generosity of Merck, there will be a reception in the auditorium as soon as or the atrium as soon as we're done. So Eric, I'll let you handle the questions.
How many networks do you typically see? You kept on using plural networks.
Right. So so I think we're early in this game where we don't yet fully understand how we organize all of this information we're seeing over the multiple tissues. But what we have been able to do is to organize these networks into these functional units. So when I say networks, I'm really referring to these, the circle thing I was showing. This is going slow right here. So we organize the information like this, where assigned to any tissue, we'll have a bunch of subnetworks, and this is sort of blurred, like the actual number is hard to nail down because these things aren't totally distinct. But we'll see within a given tissue, tens of different subnetworks that we think are reflecting interesting biological behavior. And then we'll see on the order of 10s to 20s operating between the tissues. And so over all of the tissues that we're looking at, there are literally hundreds of what I should refer to as subnetworks that collectively are making up the network that we're trying to use to predict the system. Reese?
Eric, what's the pathway to the clinic for stuff like this? how do you turn models like disease models in general into information that clinicians can use to figure out which tests need to be done, and then analyze that data to figure, to make differential diagnoses for patients?
Right, so there are two ways to use this information, right? When you have a network that's reflecting, that appears to be predictive for the disease you're looking at, you can use that network in lots of different ways. You can use that network to identify key regulators of certain modules to target for therapeutic intervention. So the path forward there would be identify those regulators, design drugs against them, and this is a game we played at Merck, and we're successful at that. For the clinical, for more of the biomarker diagnostic setting, I think what has to happen is taking some of these networks like the one I showed you for type 2 diabetes that are capturing all of that variation information, try to characterize as well as possible all the DNA, both common and rare DNA variants and environmental conditions that are affecting that, and building models based on that, and show that they outcompete the clinical models that are used to assess, say, type 2 diabetes risk. So I think the path forward is demonstrating the models have clinical utility in going beyond the sort of models that are used today to assess type 2 diabetes risk. So one of the interesting things in getting linked up with UCSF and Neil Risch is through Kaiser, you may have a path to form that kind of model, demonstrate that it's assessing, say, type 2 diabetes progression. What's the risk of give me the at-risk population at Kaiser that within a year they're at high risk of progressing to type 2 diabetes. So if we can demonstrate a model that's better at predicting that risk than the classic clinical parameters, now we can work with Kaiser to say, push this information out to your physicians. Don't tell them what to do, just say, here's your at-risk population. And now we can do a random sample of the physicians that had that information, the physicians that didn't, and then establishing whether one had a lower rate of progression than the other, and then using that as a proof of concept, a human proof of concept, to push that in more stringent trials. So that's the kind of path I'm thinking about. It is a long road. Not an easy game to play, as you know.
When you're going back to this thing about methylation or other
Yep.
aromatics, how effectively can you discriminate different modifications of DNA based simply on the pausing?
So we don't. I think the bacteria example I showed is going to blow that out and tell us how well it really can discriminate. Because what we find when we look at artificial templates, where we take the same template and we introduce different changes, is that we have near perfect ability to discriminate all the different kinds of modifications that are known. It's pretty amazing.
Simply on the kinetic.
Just based on the kinetics, the fact that it's not just affecting that one site, it has a local effect, and those local effects have much different patterns than from each other. But it's unclear to me whether that's going to generalize when we go into the wild and look at over. So it's still an open question, but it is very clear from the templates we've done that they're very different. And so you can build classifiers that very well predict which type. And then I should say that there are lots of things that we're seeing in the wild that don't look like anything that's known. So we think a lot of those are novel sort of forms of modifications that just haven't been characterized and that are just waiting to be nailed down.
You showed us a nice example in the type 2 diabetes of the mouse model being quite predictive and valuable for human, but it's just as valuable to recognize what a bad model is. So do you have any indication that networks that are being revealed in mouse are actually poor prognosticators of changes in networks and nodes that you would see in human?
Yeah, so we have many such examples. Why the hell would I show those? No. No, it is very valuable. So our bias had been sort of building up the preponderance of evidence over multiple species. So we sort of had this bias to say we want to see it, reflected in mouse, conserved into human, and so that was just how the work sort of went. But we have several sort of core disease modules in mouse that are completely non-predictive or not even seen. The structure is not similar to humans, and
that's a function of the disease model in mouse.
And that could be. It could be a disease model that's not reflected in humans.
Not necessarily.
But I think that gets
one cross, right? You have one disease, you have one
right.
diabetes model segregating.
But we do have, just to Jasper's point, we do have, not just one cross, but at Merck we generated on the order of 20 crosses, all focused on these. And so it's true, and when you look over 20 crosses that are very different and different forms of the metabolic disturbances, and you don't see some of those networks carried over into human, it is interesting. But we just haven't, it wasn't something that was driving us for the therapeutic and diagnostic benefit, but it is interesting.
Well, let's thank you
All right.
everyone one more time.
Thanks.